After thawing cells, you're now ready to plate the cell in an appropriate vessel, which could be a cell culture dish or a flask. This all depends on your specific need. For most cell types, the general accepted practice is to allow them to settle for about 24 hours after seeding. At this point, the cells should be checked for confluency, which is the percent coverage of cells over the surface of the vessel. If expanding cells, the ideal time to subculture is at 80% confluency. The time it takes to reach this level is going to vary upon cell types. Primary cells may take several days or even up to a couple of weeks to get to that point. This diagram gives a good visual of the different growth phases of cells in culture. When first plated, cells enter a lag phase where they grow slowly while trying to recover from the stress of subculturing. Cells then enter the exponential phase of growth. Here cells will grow exponentially as long as growth surface is available. When this surface is completely occupied, cells will then enter the stationary phase of no growth. If they persist in this phase, they will then enter the decline phase, in which cells will lose viability and essentially die. To ensure phenotypic stability, cell lines need to be maintained in subcultures in the exponential phase. This will need to occur before a cell monolayer becomes 100% confluent or reaches its maximum recommended cell density. So this is going to be about the ideal, again, for the subculture phase is about 80% confluency. So when cells reach 80% confluency, they can now be passaged with trypsin or ADTA. Trypsin disrupts the proteins involved in attaching cells to the culture vessel surface. The figure on the right shows the progression of cells being trypsinized. At the starting point, we see cells attached as a monolayer. After trypsin is added, the cells start to become less flat and more round. When they're completely detached, they can be seen floating around in clusters at the top of the cell culture media. This process should take about 3 to 5 minutes, but make sure to note that the dish should be agitated to help detach the cells. Long-term exposure of trypsin to cells can be detrimental. The time cells are exposed to trypsin should be minimized. Also note that if using primary cells, a special lower concentration formula of about 0.05% trypsin, 0.002% EDTA should be used. Primary cells are very sensitive to trypsins, and in this case, physical agitation of the cell culture dish is definitely needed for attachment. When trypsinization is complete, the reaction needs to be quenched, ideally with a soybean neutralizing solution. Again, this will help protect the cells from any damage of long-term exposure to trypsin.